Hello and welcome to today's fourth edition of the EANO and SNO webinar series. We're totally delighted to present you today with the highlights of the past EANO and SNO. And it's my pleasure to today moderate the session together with my co-chairs Giuseppe Minniti, Erik Sulman and Colman. And my name is, by the way, Anna Berghoff. So we have 1 hour to look over the most exciting talks and content from EANO and SNO of last year. This is not a lot of time, so we will keep it brief and do only very limited introductions of our speakers, but I thank them already for being here. Please be aware that we have a Q&A section here in the Zoom meeting, so please use this Q&A session if you have questions. Our speakers will answer questions after their talks in this Q&A, and we will have a joint but very shortly Q&A session in the end. So please use the Q&A to really get the most out of it. And with that, we start with no further ado with our first talk, and it's my pleasure to welcome Martin van den Bent, who will give us another update of the CATNON trial. Thank you, Martin, for coming.
Yeah, thank you, Anna, for the introduction. So, dear colleagues, in the next 8 minutes, I will talk you through the basic findings and the molecular findings in the EORTC CATNON trial in the subpopulation of patients with an IDH-mutated tumor. We have the next slide. Here are my disclosures. I may have the next slide. So the CATNON trial was designed in the early 2000s, prior to the discovery of the IDH mutation, to ask the question whether concurrent temozolomide and adjuvant temozolomide would improve the outcome of treatment in patients with anaplastic astrocytoma. So we designed the study basically to answer those two questions. May I have the next slide? So we had a randomization in a 2x2 design, basically asking these two questions. Next slide, please. Does concurrent temozolomide in addition to radiation therapy improve outcome? And next slide, please. And does adjuvant temozolomide given after radiation therapy do improve outcome? And in a 2x2 design, you end up with 4 treatment arms like shown here. May I have the next slide? Important to realize this was a design prior to the discovery of the IDH mutation. And of course, in today's world, the subset analysis based on the IDH mutational status is the most relevant one. May I have the next slide? So in CATNON trial, we have 444 patients with an anaplastic astrocytoma and IDH mutated. And after median follow-up of almost 11 years, 45% of the 444 patients with an IDH mutated tumor are still alive. So we have a sort of mature analysis. What I'm going to show you today is the molecular analysis. First, prognostic factor analysis looking at all the treatment arms, and then the predictive factor analysis looking at the benefit obtained with using temozolomide by comparing the 3 temozolomide arms to the patients treated with radiation therapy only. May I have the next slide? This is the overall outcome of the study. You see here the 4 treatment arms in the 444 patients with IDH mutated astrocytoma. On the right in yellow are the median survival data. May I have the next slide? As you can easily see, it's the adjuvant temozolomide that improves survival in these patients with a median survival of almost 12 years. And as you can also see is that the addition of concurrent temozolomide during radiation therapy doesn't impact outcome at all. It's really the adjuvant part of the treatment that makes a difference in the outcome of patients. With radiation therapy alone, the median survival was only 5.1 years. May I have the next slide? Today's topic is mainly the molecular analysis. We looked at DNA alterations with 2 glioma-dedicated next-generation sequencing panels, focusing on homozygous deletion of CDKN2A, PDGFR amplification, CDK4 amplification, RB status, CDK6 amplification, and PIK3CA, based on the literature alterations that have been associated with survival in other series. We also looked at methylation analysis with the in vivo methylation APIC-B chip. We looked at copy number alterations, and we used the 2 presently available classifiers to distinguish patients with an astrocytoma from an astrocytoma high grade. And we looked at MGMT with the methylation platform and with qPCR. May I have the next slide? So here you see the major molecular findings in this effort. As you can see, PDGFR amplification has a similar impact on outcome as homozygous deletion of CDKN2A. CDK4 amplification in our dataset also has an impact on outcome, but less clear. Then copy number alterations in general also has an impact on outcome. I'm showing you the data from the methylation status, MGMT promoter methylation status using the methylation array. And as you can clearly see, this did not have an impact on outcome in the patients with an IDH mutated anaplastic astrocytoma. Here's the next slide. We also looked at the diagnosis as obtained by the methylation classifier. First, I'm showing you the data from the Heidelberg classifier, the German classifier. As you can see, that classifier distinguishes between astrocytoma IDH mutated and astrocytoma IDH mutated high grade, with a clear impact on outcome. The recently developed NIH classifier, developed by Ken Aldapi and his team, may have the next slide, has a very similar prognostic significance between those two entities, the astrocytoma IDH mutated and the astrocytoma IDH mutated high grade. May have the next slide. These classifiers are nicely correlated, but not completely overlapping, as you can see here. There are cases that are differently scored by the two classifiers, but it's a minority of cases. May have the next slide. Now, of course, we did this in the hope that we could identify prognostic markers that would identify the patients that are benefiting. But to our surprise, and this is what I did not expect it, that none of the molecular markers associated with poor overall survival was predictive of lack of benefit to temozolomide. May I have the next slide? So there was benefit seen in all the poor prognostic subgroups. Astrocytoma IDH mutated high grade, as diagnosed by the classifiers, homozygous lesion of CDKN2A, and of PDGFR amplification. May I have the next slide? So here we're coming to the conclusions now of the CATNON trial. The final conclusion of the CATNON trial, there will be no further analysis. Both PDGFR amplification and homozygous lesion of CDKN2A are prognostic for outcome in astrocytoma IDH mutated. And with that, I think it's reasonable to consider PDGFR amplification as a criterion for astrocytoma IDH mutated grade 4. The methylation pattern classifiers are prognostic for overall survival within histologically defined grade 3 astrocytoma. Most of our patients had an astrocytoma IDH mutated and had an overall survival of more than 10 years. None of the highly prognostic molecular markers were predictive for outcome to temozolomide. May I have the next slide? So with a median follow-up of almost 11 years and 45% of the patients with an IDH mutated tumor still alive, we see improved overall survival after 12 cycles of adjuvant temozolomide. No benefit of concurrent temozolomide if adjuvant temozolomide is given. So the standard of care in more aggressive IDH mutated astrocytoma should be radiation therapy followed by 12 cycles of adjuvant temozolomide. May I have the next slide? And with that, I would like to acknowledge first of all, the patients that were willing to be randomized and their families for their support, and all the foundations that helped us to conduct all this translational research. And with that, I hand back the microphone to Anna.
Thank you so much, Martin. So please remember, you can ask Q&As in the Q&A session. And with that, I give over to my co-chair, Erik Sulman, for the next introduction. I think there might be a problem with unmuting, so it's...
Sorry, our next speaker will be Lusun. Apologize for that. Please go ahead. I use some time talking to myself.
Thank you, Erik. Thank you, everyone. Today, I'm very happy to present our current study of the clinical and molecular outcomes of the dual PD-1 and CTLA-4 checkpoint blockade for a recurrent glioblastoma. Next slide, please. I have no disclosures. And next slide. As we all know, glioblastoma or GBM is immunologically cold and has shown limited responsiveness to immunotherapy. And in 2019, our group, together with the Moderators team, we published paired studies in recurrent GBM patients treated with new adjuvant PD-1 blockade prior to the surgical resection. In this trial design, as you can see, one cohort of patients received PD-1 blockade before surgery, while the control group did not. After the surgery, both groups continued PD-1 blockade until disease progression. This design enabled the direct comparison of tumors at the same disease stage and provide us an unbiased view of how immunotherapy modulates the tumor microenvironment. Next slide, please. And in both the initial and extension cohort, our bulk RNA-seq analysis reviewed a distinct molecular response signature in a subset of patients treated with new adjuvant PD-1 blockade. And this signature is characterized by, next slide, please. This signature is characterized by the upregulation of T cell and interferon-related genes. And next slide, the downregulation of cell cycle and cancer proliferation genes. Importantly, this high interferon low cell cycle signature emerged as an independent positive prognostic factor for survival. And this finding confirmed the pharmacologic effect of new adjuvant PD-1 blockade, but also highlights that its clinical benefit remains confined to a small subset of patients. Next slide. Furthermore, our single-cell RNA-seq analysis of these tumors suggests that the single-agent PD-1 blockade induces adaptive immune responsiveness, including the activation of the inhibitory CTLA-4, CD86 pathways, which may paradoxically dampen the effective anti-tumor immunity. Next slide. To address this, we conducted a surgical window of opportunity trial in the same clinical setting and patient population. And in this study, we evaluated the combination of PD-1 and CTLA-4 blockade using nivolumab and ipilimumab in a randomized fashion. Next slide. Yes. And this was the actual trial design. Basically, prior to the surgical resection, patients were randomized into 3 arms. In arm 1, patients received dual ICB with EP and nivo. And in arm 2, patients received PD-1 blockade only with nivo, while patients in arm 3 received a placebo infusion. And following the surgery, patients in arms 1 and 3 received dual ICB, while arm 2 continued on nivo only until progression or toxicity. And all the enrolled patients were confirmed to be IDH wild type. So basically, this trial aimed to determine whether dual ICB could elicit more effective anti-tumor immune response compared to PD-1 blockade alone. Next slide, please. And first, we monitored the overall survival of patients across all treatment arms. Although there were no significant differences when comparing these 3 pre-surgical treatment arms, when we combined the patients who received dual ICB either before or after the surgery, which are arms 1 and 3, the median survival is 402 days compared to 276 days for PD-1 monotherapy only. Importantly, there were 5 patients achieved long-term survival. Basically, they lived more than 3 years without progression at the time of cutoff. In contrast, none of the patients who received PD-1 only achieved a long-term survival. Next slide, please. And similar to our initial PD-1 study, we collected tumor tissues from every patient in this study and performed bulk RNA sequencing. And the heat map summarized the results here. It shows the representative gene signatures that were enriched either in arm 1 or arm 2 compared to the placebo group. And as you can see, the dual ICB induced a markedly greater increase in the interferon-related gene expression compared to either the placebo or single-agent PD-1 blockade. Next slide. As you can see here, this box plot shows a representative interferon-related gene signature. It demonstrates a clear stepwise increase across the treatment groups, lowest in the placebo, intermediate with nivo only, and highest with dual blockade. Next slide. We also performed bulk TCR sequencing on these tumor samples to estimate the tumor-infiltrating lymphocyte density, TIL density. And as you can see, although the overall difference was not statistically significant, the tumors from dual ICB group did show the highest TIL density. Finally, the scatter plot on the right highlights the strong association between the TIL density and the interferon activation within the tumor microenvironment. Next slide, please. Importantly, tumor density emerged as an independent predictor of survival in this study, with higher tumor density correlating with the improved patient outcomes. Next slide. And this was also shown by this Kaplan-Meier plot. The patients with higher tumor density, they experienced longer overall survival. But when you look at this Kaplan-Meier plot more carefully, you can see that the influence of tumor density on the survival appears less pronounced among the few long-term survivors. This suggests that in addition to the intratumoral T cell abundance, other longitudinal factors likely contribute to the durable survival outcomes. Next slide. So to evaluate these longitudinal effects of treatment, we collected the blood samples at 4 time points. This includes the baseline pre-treatment, then after the neoadjuvant dosing, which is post-neo, and 2 cycles during the adjuvant phases. Again, the bulk RNA sequencing was performed on all the samples from our patients. And consistent with our intratumoral findings, we can see that patients received dual ICB before surgery showed an early increase in the interferon-related gene expression. However, despite this overall pattern at the individual level, if you can see all those lines, you can see at each patient's level, the temporal trajectories of these interferon genes, they varied a lot, even within the same treatment group. Next slide. And to capture this dynamics and the variability, we performed unsupervised clustering of this longitudinal PBMC RNA-seq profiles for each patient, and then we can review 2 distinct trajectories. And in trajectory 1, you can see there's early upregulation of interferon genes after the neoadjuvant treatment, followed by a sustained decline. While in trajectory 2, there's a delayed interferon activation, but it's persistently higher throughout the adjuvant cycles. And notably, patients in trajectory 1, which has the early interferon activation, they also had higher tumor density and stronger intratumoral interferon signaling compared to patients in trajectory 2, suggesting a potential link between systemic and intratumoral immune activation. Next slide. Interestingly, our multivariate analysis identified this early interferon upregulation as an independent predictor of prolonged survival. In contrast, patients with delayed but persistent systemic interferon activation had significantly worse outcomes, suggesting that chronic interferon and inflammatory signaling may impair the effective antitumor immunity. Next slide. So to summarize, dual PD-1 and CTLA-4 checkpoint blockade induced significantly greater tumor density and stronger intratumoral interferon activation compared to single-agent PD-1 blockade. And tumor density was strongly correlated with survival outcomes. The systemic inflammatory patterns have distinct impacts on response and survival. The early interferon upregulation followed by decline was associated with long-term survival, while the persistent interferon elevation is correlated with worse survival. So overall, these findings demonstrate a clear pharmacodynamic effect of dual ICB in recurrent glioblastoma. Next slide. So with that, I'd like to thank our collaborators and all the patients and families who participated in this study. Thank you for your attention. Okay.
Thank you so much. So we will have questions in the end, but please use the Q&A. It's my pleasure to introduce the next speaker. It's Irma Sakic from Zurich, Switzerland.
Okay, yes, thank you for the introduction. So it's now my pleasure to present you our work where we have used in vivo CRISPR screening to uncover new druggable targets in syngeneic glioblastoma mouse models. Next slide, please. So first, a little introduction about CRISPR screening. So I guess we all know that CRISPR is a really powerful approach to identify genetic dependencies. And over the years, we have learned that by increasing the complexity of the model systems, we also gain different insights to the cell-cell interactions and also the microenvironmental influences. Next slide, please. But if we now want to perform in vivo CRISPR screening, it does come with several challenges. And the most important one is the unpredictable effect during this tumor cell implantation, making it really unclear which clones successfully engraft, and also clones may be lost even before the implantation in vitro. Next slide. And to address now these limitations, we have used a novel STAR method that includes a dual guide RNA construct, which incorporates the Cre-LoxP system. So before the tumor cell implantation, all these guides are catalytically inactive, which ensures that there's no gene editing and also preserves a uniformal representation. And only after the addition of tamoxifen, the Cre recombination is then induced, which then generates either an active or an inactive guide RNA version. So on the left, you see an active guide RNA, which is capable of inducing a gene knockout, which then couples to Cas9. Or then the inactive guide RNA, which then is not able to introduce a genetic knockout and only serves as an internal control for us. We're staying on the slide. So after the tumor cell implantation, both of these guide RNAs remain still detectable in the tumor. And if now an active guide RNA targets an essential gene, their number will drop over time, while guides to target tumor-promoting genes, these will enrich. And this design now allows us to distinguish between true biological effects and random loss. And finally, for our data interpretation, we then compare these ratios of active and inactive guide RNAs, where a negative ratio indicates the downregulation and thereby the identification of a genetic vulnerability. Next slide, please. So in the end, we have performed first a genome-wide screen, and building on the results, we have designed a more targeted, smaller in vivo library. Both in a syngeneic orthotopic glioma model with the immunotherapeutic pressure using a tumor-targeting cytokine L19 TNF. Next slide. Thank you. And based on the results, we have seen that in the genome-wide screen, we had a really strong gene-level representation, shown in green. And by reducing the complexity of our library, we have also further improved the guide RNA coverage, shown in violet. And then the key strength was really in the robustness of our approach, where I show you in the right corner only the top ranking genes, but where we have observed a consistent guide RNA dropout pattern, which really highlights the reproducibility of these signals across our different screens. Next slide, please. We also have looked at the quality of the screen by observing that the control genes that we incorporated, including known essential genes shown in red, that they drop, and the non-essential genes shown in white, they cluster around 0. And this together indicates a strong sensitivity of our system. And then finally, to identify a target gene, we performed comparative analysis of the screen results between the targeted and the control group, and it revealed the DDX39B as the most strongly statistically significant depleting HICT, which was also backed up by multiple guide RNAs in both the control and the treatment group. And this gene encodes a dead box RNA helicase, which is a core component of the spliceosome, with a key role in mRNA splicing and also mRNA export into the cytoplasm. Next slide, please. So, of course, we also investigated whether this gene now was also relevant in human glioblastoma, and we analyzed multiple independent transcriptomic data sets from human glioblastoma cohorts, and we found that the DDX was predominantly expressed in glioma tissue with minimal expression in the immune compartments. And spatial transcriptomic analysis further showed that the expression was highest in proliferative OPC-like glioma subpopulations. And additionally, a higher median expression was also associated with poorer overall survival. Next slide, please. And also we had in-house data sets of matched newly diagnosed and recurrent samples that also showed a higher expression level within the tumor region compared to the adjacent stroma. Next slide, please. And we also have quantified these data where we observed a higher expression within the tumor region compared to stroma, and also that the expression intensity was comparable between the newly diagnosed and the recurrent patients. So together, these patterns suggested that DDX39B potentially could be also used to target pharmacologically. Next slide, please. And we investigated that, and we have found a small molecule that targets the catalytic function of the protein. And in vitro, we observed a robust cell death upon the inhibition. Next slide, please. So we also wanted to gain a bit more mechanistic insight. So we performed proteomic analysis together with a colleague of mine, Adela. And we have found that a negative enrichment of RNA metabolism pathways, which also reflects now the pattern that is consistent with the known role of DDX in mRNA splicing, but also the export. And this actually also supports the target disruption of the RNA homeostasis following this pharmacological inhibition. And additionally, we also observed positive enrichment of pathways related to metabolic adaptation and redox regulation. Next slide, please. And to assess now how DDX inhibition also affects the cell fate, because we observed cell death in vitro, we examined apoptotic signaling at the protein level, and we have observed a downregulation of key regulators of DNA damage tolerance and also anti-apoptotic signaling. Next slide, please. And we also observed pro-apoptotic proteins that were upregulated. And also upon the strongest differentially expressed proteins, we found that these were also associated with the redox imbalance response, indicating that upon the DDX inhibition, the cells react in a protective answer against oxidative stress. So together, these patterns reflect a shift from survival and damage tolerance toward apoptotic commitment, indicating that DDX inhibition lowers the apoptotic threshold in glioblastoma cells. Yes, perfect. Thank you. And also we extended these findings then also in vivo again, where we used pharmacological inhibition of this protein together with the immunocytokine therapy L19TNF. And we observed a prolonged survival and even a one long-time survivor in the combination treatment. But overall, there was no observed synergistic effect in this CT2A model, which is known to be a rather immune therapy resistant. So currently, next slide, we are looking into other glioblastoma models that are more responsive to immunotherapy to investigate whether there might be a synergistic or additive effect of combining DDX inhibition together with L19TNF. And also we are now currently evaluating our glioblastoma ex vivo patient samples where we treated dose suspensions with the DDX inhibitor to investigate whether we observe there also is cancer cell killing. So in the end, next slide, I'm already at the end, and I quickly want to acknowledge all the lab members and also collaborators, and thank you for listening.
Thank you. I give over to my co-chair, Howard Colman, to introduce the next speaker.
Thank you. Our next speaker is Nandina Acharya from Ohio State University.
Thank you so much for the introduction. So today I will be talking to you about how glioblastoma hijacks the sensorineural circuit to suppress immune responses and how we can rewire these neuroimmune interactions to lead to immunotherapy sensitivity in glioblastoma. So glioblastoma is the most aggressive primary brain tumor, and the survival in this case is one of the most worst compared to different types of solid cancers. So this leads us to the important question. Next slide, please. Why is glioblastoma so resistant to immunotherapy? We heard several talks about this, but I'll quickly summarize. The most important reasons why it is resistant to immunotherapy is because of the massive heterogeneity, both intertumoral and intratumoral, the profound immune suppressive microenvironment, mostly generated by the myeloid cells, and also the unique anatomical location of this tumor type. It is present inside the brain, which is a very immune distinct organ, where immune responses are highly nuanced. So all these factors collectively generate an ecosystem which limits immune surveillance and also blunts anti-tumor immune responses. So for us to move the field forward, it's important to recognize very unique anatomical regulators of immune responses. Next slide, please. As I mentioned, the brain is immunologically distinct. What I mean by that is there are very, very specialized niches which regulate immune responses. And today we will be focusing on the meninges. Next slide, please. The meninges is a very important neuroimmune interface. As you can see here in the whole mount image, it is highly innervated by the CGRP positive sensory neurons. In the last couple of years, we've seen a lot of work suggesting that the cortical neurons can regulate the pathophysiology of GBM. However, the role of these sensory neurons in GBM has not been explored. Next slide, please. These nociceptors, which are a subset of sensory neuron, is traditionally associated with pain transmission, but we also know that they can regulate immune responses and move the immune responses from anti-inflammatory to pro-inflammatory depending upon the context. Next slide, please. So we asked the question, does GBM engage these nociceptors? To answer that question, we harvested the trigeminal ganglia, which houses the cell bodies of these neurons, and then we looked for markers of activation, mostly the CGRP, the neuropeptide, and CFOS. And compared to the sham-operated, we see that in different models of GBM, the TG nociceptors were activated. Next slide, please. We also measured the amount of this neuropeptide release, and in different models of GBM, we do see that these nociceptors lead to, the nociceptors from GBM-bearing mice lead to higher production of CGRP. So this shows that in GBM, nociceptors are activated. Next slide, please. So because these trigeminal ganglia are outside the brain parenchyma, we hypothesized that perhaps there are some soluble mediators which are secreted by the tumor that could be activating these neurons. So to test that, we did the simple experiment where we took naive trigeminal ganglia and we cultured them with CSF derived from either sham-operated mice or from GBM-bearing mice. And as you can see in the images here, the CSF from GBM-bearing mice did lead to striking neuronal outgrowth, increased calcium transit, and also increased production of CGRP. So these show that the tumors actively produce metabolites into the CSF that can lead to the activation of these neurons. Next slide, please. So we wanted to see the functional role of these neurons in tumor progression. So for that, we ablated the nociceptors using this chemical called RTX, and then we implanted tumors to these mice. A cohort of mice also got immunotherapy. Next slide, please. And what we found here is that treatment with RTX, so basically ablating nociceptors, did lead to improved survival, as you can see in the green line. But what was more dramatic is that when you ablate these nociceptors, the GBM-bearing mice now become sensitive to immunotherapy. So this shows that these nociceptors do contribute to resistance of GBM to immunotherapy. And if you can target these nociceptors, you unleash the sensitivity. Next slide, please. In a complementary experiment, we did pharmacological silencing of these nociceptors, and here too we see that the survival did improve. Next slide, please. So overall, in this first part, I'll quickly summarize that we saw that tumors do produce some certain metabolites into the CSF that can be carried to the trigeminal ganglia, which is outside of the brain parenchyma, and activates the nociceptors there. And these nociceptors have axonal fibers in the meninges, which can produce neuropeptides, which could potentially affect immune responses. So the next question we asked is, do these neuropeptides produced by these hyperactivated nociceptors have any bearing on immune regulation in GBM? Next slide, please. So to answer this, we conducted a simple in vitro experiment. We harvested the TGs from sham-operated or GBM-bearing mice, and then we derived the conditioned media. We applied that conditioned media to bone marrow-derived macrophages. As you can see in the top panel, the conditioned media from the GBM TGs led to expression of arginase 1, which is a hallmark immune suppressive marker. But importantly, it was very important that this effect was completely abrogated when the macrophages lacked the expression of RAMP1. RAMP1 is the receptor for CGRP, showing that this is all driven by the neuropeptide CGRP. Next slide, please. So we wanted to see if we ablate these sensory neuron pathways of CGRP in vivo, does it have any bearing on tumor growth? So we generated these myeloid knockouts where RAMP1, the receptor of CGRP, is ablated. And as you can see in the green line, the knockout did survive longer than the wild type. And importantly, it also led to increased sensitivity to immunotherapy. Also, another critical finding was that this effect in the knockout, the survival benefit, was completely abrogated when the CD8 T cells were depleted. Next slide, please. We wanted to phenotype the macrophages in these knockout more, and we saw that when we removed RAMP1, or basically CGRP signaling, the macrophages were shifted more from an anti-inflammatory phenotype to more pro-stimulatory phenotype, and they were better antigen-presenting cells. When we transcriptionally look at these macrophages, we saw that the knockout mice had better antigen processing and presenting signature, and functionally, when we co-cultured the macrophages, that were treated with CGRP, with CD8 T cells, we saw that the CD8 T cells that was co-cultured with macrophages treated with CGRP had lower proliferation. So this shows that CGRP ramp1 pathway constrains the antigen presenting ability and T cell stimulatory capacity of these macrophages. Next slide, please. We wanted to see how does this pathway of nociceptors on macrophages interact with other immune pathways on macrophages. So we looked at the macrophages which were derived from mice treated with CGRP antagonists, and we found that there was a profound enrichment for interferon gamma pathways. We also looked in human GBM dataset, and we saw that the interferon gamma and CGRP pathways are negatively correlated. Next slide, please. Mechanistically, we found that interferon gamma dramatically reduced the expression of RAMP1, the receptor for CGRP. Next slide, please. Also, on the other hand, CGRP led to reduction in the frequency of phosphostat1, which is downstream of interferon gamma. So basically, CGRP and interferon gamma are mutually antagonistic, and they can fine-tune the fate of macrophages in the tumor microenvironment. Next slide, please. Then finally, because these knockout mice were on myeloid cells, we wanted to see the impact on CD8 T cells. We found that there was high infiltration of CD8 T cells in the tumor microenvironment of the knockout mice. They had better effector capacity, as you can see with perforin and granzyme. Next slide, please. And when we looked at the CD8 PD-1 positive fraction, we saw that there was an increased abundance of stem-like cells, the TCF1 positive cells, and reduced amount of the TIM3 positive dysfunctional cells. Next slide, please. We profiled these TIM3 positive cells further, and we found that in the knockout, it produced higher amounts of cytokine. And when we looked at the other parameters of dysfunction or exhaustion, we saw that the knockout had lesser severity of exhaustion. Next slide, please. So overall, what I showed you today is that GBM activates nociceptors, and they produce higher amounts of this neuropeptide CGRP, which can act on the macrophages and make them more pro-tumorigenic or anti-inflammatory. And it does this by inhibiting the function of interferon gamma. However, if you abrogate this pathway, it unleashes the antigen-presenting ability of the macrophages, and they become better stimulators of CD8 T cells, leading to improved CD8 T cell function, improved anti-tumor immune responses, and most importantly, it sensitizes GBM to immunotherapy. Next slide, please. So with that, I thank you all for your kind attention, and I would like to thank all the lab members and collaborators. Thank you so much.
Thank you so much. So we're moving on to our next speaker, Daruri Jin from Heidelberg, Germany.
Thank you, Anna, for the introduction. So one thing before my talk, please excuse the dressing on my face because I had a small bike accident the other day, so I hope this won't distract you from my presentation. So next slide, please. So in the next few minutes, I will walk you through our work, AI-based histopathological classification of CNS tumors. And actually, like now we modify this title a bit. Now it's histology-based AI prediction of CNS tumor methylation subtype. So just for your information. So in recent years, artificial intelligence has really transformed the digital pathology domain. It has made significant progress in the aspect of diagnosis, detection, and segmentation, molecular profile prediction, and prognosis. This progress sets a very solid foundation for our work on CNS tumor analysis. Next slide, please. But first, let's look at the current diagnostic standard. In the clinic, we usually combine the result from histology examination, IHC, and DNA methylation tests. So DNA methylation profiling is especially important because it provides a very fine-grained classification of the tumors that aligns better with the clinical outcomes. But at the same time, we all know that it will definitely require additional resources and significantly extend the turnaround time, so which could take 2 to 3 weeks to get the final result. So we ask ourselves, could AI improve this workflow, and can we directly predict the methylation classes from the H&E slides, which are more rapidly and widely available? Next slide, please. So here is our proposed pipeline named Heteros, which means companion in Greek. So we'll have like a simplified version showing the inference process here. We will first detect the tissue region and tile them into small patches. So this is the pre-processing of the slides. And then we will further extract the eye embedding through a pre-trained vision transformer named Prof Gigapath, so which is also a pathology foundation model, and use a constant multiple instance learning framework to compute the slide level representation and the final 102 class predictions. Next slide, please. So for training and validating heteros, we actually compile very large and diverse cohort. So it's around like 11K slides scattered from 10 centers across 4 continents. These slides covered in a range of 402 CNS tumor classes. Next slide, please. So in internal validation, we found the heteros confidence really match up with the actual accuracy. This is a very important feature for real-world usage because it basically tells you when and how you can trust the model. So for example, here we set the confidence threshold as 0.5. Next slide. Next slide, please. Yeah, so we will get like 70% high confidence prediction and remaining 30% low confidence ones. And as you can see from the Sankey plot here, the high confidence group is very reliable with a top one accuracy of 88%. Next slide, please. For the low confidence group, it drops to 46% as we expected. The top one here means we only look at the prediction with the highest predicted probability from the model. Next slide, please. But the top one prediction may be like too strict. So if we look at the top 3 accuracy, even low confidence group hits 71%. So this means heteros could meaningfully narrow down the possibilities from 102 classes to 3 classes, which is a huge help in clinical settings. Next slide, please. And we'll validate heteros with a very comprehensive external data set comprising 9 institutes from 6 countries. And we also observed a very similar accuracy in different confidence groups compared to our internal validation result. Next slide, please. We also conducted an interesting comparison between heteros and 5 board-certified neuropathologists based on a small cohort of 210 samples. So the leftmost column is the result from heteros. Next slide, please. And the others are from human evaluators. So basically, it shows heteros consistently outperform human evaluators across all heteros confidence intervals with an average accuracy of 68% against 28%. Next slide, please. And then you might wonder like how this actually helps in the real world application. So one of the biggest advantages here is the output heat maps. So it highlights the most informative region, which will let pathologists to review heteros decision-making process and also guide the selection of the tissue for further molecular testing. Next slide, please. Next. And next. To assess the potential clinical utility of heteros, the algorithm was prospectively evaluated alongside the routine diagnostics at the Department of Neuropathology, Heidelberg. This slide only shows like the sample going with the methylation test. For routine diagnosis, apparently it takes around 2 weeks to get the methylation result, while heteros ran on a consumer-grade hardware, taking an average of 10 minutes to process a slide and generated the corresponding reports. So 2/3 of the prediction are predicted with a high confidence and also achieved an accuracy of 94% for the cases with high methylation scores. And next slide, please. If we look into the prediction with the low methylation scores, we find that heteros accuracy is actually slightly higher than the methylation profiling itself when compared against the integrated diagnosis. And if heteros could agree with the like the diagnosis with low methylation scores, the accuracy is nearly 90%. So overall, this result shows Hetaro's potential to reduce the tie significantly while maintaining good accuracy. And for the next step, we will keep pushing to increase the diversity of the training set to further improve the model's generalization. Next slide, please. Thank you for the listening, and huge thanks to everyone from Gerstner Lab, the team at Neuropathology Uniclinic, and my colleagues at Beihang University for their support. Thank you.
Thank
you.
you.
For our last speaker, we'll have Christopher Dampier present, please.
Thank you. I'm Chris Dampier. I'm a fellow, neuropathology fellow at Johns Hopkins and the National Cancer Institute. Great, thank you. So I'm honored to have the opportunity to talk about NeuroPath AI, a deep neural network for CNS tumor classification from digital whole slide images. I'm presenting on behalf of myself and Lao Chung and Dr. Aldape, others who contributed to this work. Next slide, please. The problem we're trying, well, so I want to make three key points over the next 8 minutes. First is that deep neural networks can infer molecular features from histopathology images. The second is that inferred molecular features can help predict tumor types from histopathology images. And the third is that AI assistants can help humans classify brain tumors from histopathology images. Next slide, please. The problem we're trying to address here is that classification systems for CNS tumors are complex and dynamic. Here's an example chapter from the 5th edition WHO CNS classification that lists lots of different glial and glial-neuronal tumor types, 2 of which require DNA methylation profiling to diagnose. And so this can be challenging for a human neuropathologist. Next slide, please. A related problem is that new molecularly distinct entities often look like old ones. And so here are 2 example microscopy images of glial tumors with perivascular pseudorosettes. And from this microscopy, it's hard to know which one is likely to harbor a ZFTA fusion or a PLAG L1 fusion or a PAT-Z1 fusion. Next slide, please. But we believe computational pathology can help. And so this is a schematic overview of our algorithms architecture. Next slide, please. The first step is that we convert whole slide images into numbers and an abstract set of features that can be summarized across different types of slides. Next slide, please. And we use a first model that classifies tumors directly from the image features. Next slide, please. We then use the image features to infer DNA methylation profile and gene expression profile, and then use these inferred molecular features to classify the tumors. Next slide, please. A 4th submodule classifies tumors based on the tumor location and then the patient age and sex. Next slide, please. The outputs from these 4 submodules are integrated in a separate module that arrives at a diagnosis. When this diagnosis can be refined further, next slide, please, we implement a recursive step that goes through the process again until a terminal classification can be reached. Next slide, please. The neural networks that are used to infer the molecular features were first introduced by the Aldape lab and others. Next slide, please. In this publication that showed that inferred DNA methylation patterns can be used to classify CNS tumors. And so our work builds on this. Next slide, please. And we apply this also to the RNA expression module that infers RNA expression from histopathology and uses this expression pattern to classify brain tumors. Next slide, please. But for the classification component of this algorithm, we started with a large training set across some different centers in North America and Europe, the Digital Brain Tumor Atlas being from Europe. And we include about 5,800 tumor samples. Next slide, please. And then we test the algorithm on another large data set of about 5,500 patient samples collected from across a few centers in North America and one in the UK. All of these tumors have high confidence DNA methylation based tumor classification labels as our ground truth. Next slide, please. When we test the algorithm, well, first of all, I should note that we try to simplify the problem of classifying brain tumors by breaking the complex problem into multiple simpler problems. Each of these nested classifiers that we include in our classification model includes no more than 9 tumor types that must be predicted at any given stage of the classification. And of the 18 nested classifiers that we incorporate, they're able to predict 52 clinically relevant tumor types, 41 of which are WHO entities. Next slide, please. So when we test the algorithm in the test cohort, we find that 87% of the test samples receive an interpretable algorithm prediction. And shown here is the simple accuracy across the different submodules. And the point here is that the integrated submodule gives the highest simple accuracy, underscoring the utility of incorporating these inferred molecular features into the classification schema. Next slide, please. We envision this algorithm being utilized by pathologists prior to assessment as part of the processing workflow. The H&E comes off the stainer, gets digitized, passed through the algorithm, and then a neuropathologist is able to incorporate the AI predictions at the time of their differential diagnosis generation so that they can tailor the immunohistochemical and molecular studies, the ancillary studies, to reach an integrated histomolecular diagnosis more efficiently. Next slide, please. In this context, the relevant outputs from the algorithm, not necessarily just the top scoring output, but the top 2 to generate a differential diagnosis for the human pathologist. And that's the accuracy of the top 2 predictions is shown in gray when you consider the top 2. Now, a major caveat is that we have class imbalance in our test set. And so balanced accuracy is really the better metric to consider the utility of these predictions. And we see that 75% balanced accuracy for the top 2 predictions is achieved by our algorithm. And we think that's useful. Next slide, please. But another way of looking at the utility of the algorithm is to compare it to human neuropathologists. And so we asked 8 human neuropathologists to predict tumor types from whole slide images, just like the algorithm is doing. And we see that the accuracy of the algorithm compared favorably to the human pathologists in a subset of the NCI samples, which are enriched for challenging cases. And so we thought this was good evidence that the algorithm was a useful tool. Next slide, please. But we expanded on this to ask, and we asked the human neuropathologist to reevaluate each of the whole slide images with access to the predictions from NeuroPath AI algorithm. And on the left, I'm showing that accuracy for all of the human neuropathologists increased when they had no AI versus when AI assistance was provided. The slopes of all these lines are positive, suggesting that even the highest performing human neuropathologists can benefit from the AI. In the middle, I'm showing the mean accuracy per sample is statistically significantly better when AI assistance is provided to human neuropathologists. So if it's your tumor as a patient, you would like to have AI assistance available to your neuropathologist for initial assessment of your tumor. And then on the right, I'm showing that the interhuman concordance is improved when AI assistance is available to them. Next slide, please. We can also imagine a scenario in which ancillary testing, access to ancillary testing is limited. And so in this context, you could implement stringent confidence score cutoffs for the algorithm predictions. And while this will result in lower coverage of the samples that you're testing, you will achieve high accuracy with these stringent confidence score cutoffs. Next slide, please. And in the subset of samples that we compared with human neuropathologists that reached high confidence predictions from NeuroPath AI, we see that no humans approached the algorithm in terms of accuracy. Next slide, please. The algorithm is, we want to make it available through our website. This is the same website that the NCI has available for IDAT, for methylation-based classification upload of IDAT files. There's a new tab that we've added that through which you can upload whole slide images. At this point, only family level classifications are available. It's somewhat slowed down by some recent stress at the NIH, not unlike the NATO alliance. And so we're trying to work through some of those challenges, and we hope to have terminal classifications available through the website at some point soon. Next slide, please. This is my acknowledgement slide, especially Dr. Dape and Lao Chung. And next slide, please. I'll just leave up the website for a second. This is where we'd like to be able to receive your whole slide images to produce useful classifications in the future. Thank you.
Thank you so much, and thanks everybody for staying in time. So we have time for some burning questions, and I ask my co-chairs to also help me here with the Q&A. I think the first burning question goes to the direction of Martin van den Bent, because there were quite a few questions in the direction. After seeing your data of today, how should we treat an astrocytoma, IDH mutant grade 4? Is that how treated as a glioblastoma, or is it treated after the CATNON protocol? And what are your concerns and thoughts here?
Well, it's a question that keeps coming back and back. And the first thing to be clearly understood is that glioblastoma is a different disease than IDH-mutated glioma. The IDH mutation brings a lot of vulnerabilities for treatment that the glioblastoma simply doesn't have. That's one thing. What we showed in the CATNON trial is that the concurrent phase of the treatment doesn't make an impact. And what we also showed in the CATNON trial, because we were using the WHO 2007, we have in the CATNON trial patients with an outcome that reflects astrocytoma grade 4. So the only data that we have today about the treatment of grade 4 astrocytoma stems from the CATNON trial, not from a glioblastoma trial. That's a different disease. You cannot compare trials on pancreatic cancer with renal cancer just because they are in the abdomen.
Thanks. So I hear that you would strongly argue it's 12 cycles adjuvant and no concurrent temozolomide.
Yes, that's my current position on this. And this is the only data that we have, basically. There are no other data.
I couldn't agree more. I thank you so much for pointing that out to our audience, because I think it's still an area of discussion. Howard and Erik, some questions from your side that you thought would be burning?
So there was a question for Lussan about TIL density and looks like maybe first diagnosis versus recurrence.
Yeah, unfortunately, we don't have that data. We don't have the tumor, so we cannot provide the tumor. But as I answered in the chat box, we did have the PBMC data pre-treatment, and there's a tendency that if the patients had TIL density higher, then they will also have PBMC enriched with a T cell signature. And this T cell signature is specifically a stem-like or progenitor-like T cell signature. So meaning these patients are poised for like productive anti-tumor immunity. So this could be a guidance for us to select the patients for specific treatments.
Thank you. And there was a question, I think, to Irma on a migraine. No, I'm sorry, I forgot to Nandini on migraine treatment. I think that's very interesting direction. Maybe you can comment whether you think there is a clinical application of your findings.
Yeah, this is a very important question. We have tested those drugs, but we don't see a very clear result in that. It depends upon the model. And that is because CGRP, this neuropeptide, it's known to have diverse effect on different immune cells. Like it can be immune suppressive on macrophages, but it's immune stimulatory on DCs, for example. So if you curtail the pathway altogether, the net effect, we don't see an improvement in survival. But like, again, I said, if it's an immunogenic tumor, we do see improvement in survival. If it's a very cold tumor, we don't. So we are, investigating at this current moment what could be the underlying reason for it. And then we're also trying to make some bispecific antibodies to just curtail this pathway on macrophages, because the data of abrogating this pathway on macrophages is very clear. You do lead to improved sensitivity to GBM. So, yeah.
Thanks. One question to Christopher from my side. I think you nicely pointed out how important it is to use AI. So which or who pathologists do you think should use these AI tools? Everybody or only selected ones?
Yeah. So this is a great question with respect to, CNS tumor diagnosis is often performed by neuropathologists, but a lot of resections, especially more common, meningiomas, glioblastomas, are often seen at a center that doesn't have neuropathologists. So while we've focused on showing how our tools can be helpful to neuropathologists, I think the tool would be even more useful to generalists or surgical pathologists who face these tumors without, specialist access, or at least ready access to specialists.
Thank you. We have time for one more burning question. Howard or Erik, from your side, do you think that there is something that we should discuss?
There was one new one in the chat. This is for Dr. Dampier. For the two AI-based pathology classification algorithms, can the two algorithms improve sensitivity and specificity with more samples? Are they ready for clinical use?
Yeah, the excellent question. Thank you. This is something we're exploring right now. We have, for our next, iteration of the model, we think by including more. So we've doubled the training set now, and we think that it will produce a better classification algorithm. And this is unproven, but it's something that we're working on. And yeah, ultimately, we'd like to put it in a context where it can be evaluated prospectively, like at centers that have digital pathology. I think that, many might be interested in evaluating either of these algorithms in their workflow. And I think both could be proved to be useful, but it's not yet proven.
Thank you so much. So we used up all our time. Also, I feel that there would be more discussion if possible. Thank to all the speakers. Thank for my co-chairs, and particularly thank you to all in the audience for the interest and the interesting questions. And I hope to see you at EANO and/or SNO this year, and at the latest then at our webinar that we will host again in 1 year from now. Have a great evening.